In this video, we'll look at the Antibody Sequence Liabilities data function in ChemCharts. The Antibody Sequence Liabilities data function identifies sequences that could be potentially problematic during antibody development. The minimum input needed is an antibody sequence, but if structural information is available, the data function will only consider residues that are solvent exposed. In this example, I have 10 antibody sequences for which structures have been predicted with the antibody structure prediction data function. The relative accessible surface area data function has also been run on the predicted structures to provide information about residue solvent accessibility. In the data function settings dialog, select the data table and the column containing the sequences, and provide a name for the output column. By default, all available sequence liability searches are selected. Liabilities can be removed by clicking the X next to the liability name. Clicking in the selection box shows a list of available liabilities which can be selected or unselected by clicking on the checkbox to the left. Definitions of the liabilities are shown in this table from the ChemCharts Data Function User's Guide. Annotations showing the location and details of the liabilities will be created in the output column. Select a color for the annotation. When the data function is finished, a new column is created with annotations indicating the location of any liabilities found. Hovering over the annotation shows a pop-up window with more information about that specific liability. Additional columns are also created for each liability with true or false if the liability was found or not. These columns can be used with filters for quick evaluation of antibody sequences. When structures are available, the Biomolecule viewer provides a powerful and convenient interface to explore the identified liabilities in 3D. Here I have the Biomolecule viewer and a table visual. The viewer has been configured to display the predicted structures, and the sequence pane displays the column containing sequences annotated with antibody numbering, RASA, and sequence liabilities. You may need to expand the rows in the sequence pane to see all of the annotations. Hovering over an annotation shows a pop-up window with additional information about that annotation. Clicking on an annotation will focus the viewer on that residue and highlight it. Thanks for watching, and check out our other ChemCharts data function tutorials.